And I really want to thank the organizers for inviting me. It's quite nice to be at this meeting, and it's very nice to go after Andy and before, who's my advisor, postdoc advisor, and before Glenn, who was his advisor. What I'm going to talk about are things that relate to social diversity, primarily in birds, but not exclusively, and some aspects of cognition that might go along with that social diversity. This is kind of a general question that I've been interested in a long time, since working at Andy's lab and even before that, asking about what kinds of things are deeply conserved. Can we identify some common themes across all vertebrates? Today I'm going to focus more on this, is when you take a look at those networks, how do they get tweaked evolutionarily to produce divergence in behavior? And this kind of revisits some of Paul's themes from earlier. And when we see homoplastic behavioral developments or evolution, do we see convergence or parallel evolution in the underlying neural mechanisms? So one of the kind of scary things that you see in social neuroscience is the desire to use model systems, a single model system for some aspect of social behavior that is really evolutionarily labile. There are all kinds of things about social behavior that are very evolutionarily labile, that are very important, of great interest, things that I particularly find interesting: mating systems, cooperative breeding, patterns of grouping, which is what we're going to talk about most today. But you can't really generate any idea of what the general principles are, the most commonly hit upon solutions to these things are, without taking a comparative approach. We're going to talk about birds today, but as I'll point out, there are some similarities to humans. And I think everybody in here knows this, is that independent evolution of a particular aspect of behavior can be achieved by selection on different kinds of neural mechanisms. So I'm going to start off today by talking about grouping behavior in a strilded finch species, not this particular one, but in a strilded finch species that exhibit fairly stable forms of sociality year-round, independent of context. And then we're going to take ideas from that towards the end of the talk and see if we can predict what kinds of changes are going on seasonally in animals that go from being territorial to flocking during the winter. So grouping is an incredibly important dimension of social structure. I probably don't have to belabor that point. It has broad-ranging impacts on all kinds of other behaviors, physiology, disease transmission, etc. But large-scale variation in grouping has been virtually ignored by neurobiologists, and there are probably a couple of really good reasons for that. Number one, it's hard to identify related species of rodents that you can study easily in the lab that exhibit large-scale variation in grouping. But even among rodents, if you were to pick from all the rodents that are out there in the world, when you find differences in patterns of grouping, the species often tend to differ in lots of other ways. Maybe it's patterns of parental care, maybe it's mating system, maybe it's aspects of their ecology that are going to impact neuroendocrine systems, and of course you don't have a dedicated system probably for grouping and parental care and pair bonding. These things are probably sharing a lot of mechanisms, so you want to control for these other things. You might be able to do it in ungulates, and my program officer has assured me that she has wildebeest on her wish list for me, but I haven't gotten them yet. What we're going to talk about today then are these questions. What are the mechanisms that titrate group size preferences, and how do those mechanisms evolve when we're holding other things constant? Those other things likely will influence how neural mechanisms of grouping can evolve. Mating system, patterns of parental care, other aspects of ecology that require some of the same mechanisms for physiological regulation, things like that. So what we're going to do today is scratch the surface. We're going to get some ideas of where to start, and then we have to look at how things evolve on different backgrounds from here. So I chose the Astrildid finch family for a couple of reasons, but they're really good for looking at this. And this is the big reason why, is that you can isolate species-typical group size as a quasi-experimental variable. This is something that you can't do in most vertebrate groups. And there are very few bird families that allow you to do this. There are about 140 species of Australid finches. All of those are monogamous to the extent that we know, and to the extent we know, most of them are lifelong pair bonders. They all exhibit biparental care. We've been working with 5 species of those, so this doesn't characterize all Australids, just the 5 species that we've been working with, and they live and breed in semi-arid or grassland scrub habitat, breed semi-opportunistically dependent upon rainfall. And I already made this point, but it's very important. There's minimal conditional variation. It doesn't matter what time of year it is, what the context is. The more social species are always more social. The less social species are always less social. This is one of those species here, an extremely aggressive finch, but very tight pair bonds. The male went in first and did his partner the kindness of calling her into the net. Another advantage of working in the Australids is that we have zebra finches in that family, and as I imagine you're aware, there are all kinds of genomic resources for zebra finches now, plus their behavior is just incredibly robust in the lab. You can get wild-type behavior, complex social behavior in the lab. It's readily observable, readily quantifiable, and they're also very good. I'm NIH funded. They're very good for comparing to people because they use sensory systems in much the same way that we do. They're primarily visual, primarily vocal. They have biparental nuclear families that are embedded in larger social groups, etc. So there are a lot of things about them. Even though they're phylogenetically distantly related, there are some good reasons to focus on them. So we're working with 2 species that independently evolved a really highly gregarious form of social structure. They live in large groups during both the breeding season and non-breeding season. The average group size is maybe about 100. Zebra finches you'll find sometimes in groups of 300 or more. Spice finches, at times you'll find in groups of, thousands, 3,000. I've been in a group of spice finches that I couldn't even find the end of before, just hiking, and they seem to never end. And I'll hit this point about independent evolution again in a minute. We have independent evolution at the other end of the continuum too. The Melba finch here and this homely critter, the violet-eared waxbill. These evolved social structure of territoriality, and I said year-round asociality. They live as male-female pairs year-round, and of course they have dependent young for part of the year. As you'll see in a second, the violet-eared waxbill, which I have listed as Urea ginthus granatina, has been put in a different genus by Sorensen, but there's not a species-level phylogeny yet, so I keep it in Urea ginthus. But whatever the case, it's closely related to this species here, and this species is sympatric with both of the territorial species that I just showed you, but this species lives in modest-sized groups during the non-breeding season. They loosely distribute for breeding, but you'll find adults out foraging together. So everybody just kind of finds their own space to breed, but they don't get territorial, and they continue to maintain social contact with other adults. This beautiful little animal here is typical of most of the 140 Australid species that are out there. They form small parties when they're not breeding. These birds were in a group of 5 when they were photographed. They loosely distribute for breeding without being territorial. So the fact that that is a very, very common pattern in the family gives us the opportunity to readily identify cases of independent evolution of this extreme gregariousness or of territoriality. So you can ignore these down here. These are the Australid genera here, based on some work by Sorensen. The red down here highlights the genera in which this high level of gregariousness has evolved. Taenia pygia, which is the zebra finch, and a species that used to be considered a subspecies of zebra finch, the chestnut-eared finch, and then 3 of the 26 Lonchura species down here have evolved that kind of social structure. And I need to emphasize, these guys are off the charts relative to the next most social species in this family. So you do find other species that breed colonially, but it might be 5 pairs, maximum up to 10. So it's like a switch goes off in these particular genera, and you get species that are just way off the charts relative to everybody else. Up here you see cases where territoriality is involved independently. This is a zoom in of that. The Melba finch is one of 5 species in Pytilia. It's the only territorial species in that family. The others are typical Astrilidids. These outgroups are typical Astrilidids, so they are not territorial and they form small parties during the non-breeding season. The violet-eared waxbill is here. The Ureaginthus species listed here, the blue waxbills, are more gregarious than the average Astrilidids. The outgroups here are typical Astrilidids. So independent evolution at both ends of the social spectrum. So where are we going to look in the brain then to figure out what's titrating the group size preferences of these birds? This is just a multi-labeled section through the middle of a zebrafinch brain, and the labels don't matter so much. It just helps you see some areas. Anterior commissures here, lateral ventricles is here. Variety of these hypothalamic areas, lateral septum, bed nucleus are areas that are shown here. And this is simply meant to show that there are reciprocal connections throughout a suite of hypothalamic, midbrain areas, lateral septum, medial extended amygdala. The connections are certainly stronger in some directions than others. That's not shown here. These are certainly not dedicated structures. They aren't dedicated to social behavior, but collectively they are absolutely essential for the regulation of these behaviors listed over here. And when I say essential, if you start taking these structures out, animals just will not do the behaviors. They are absolutely required for these behaviors, and this has been shown in a large body of work. This diagram's adapted from a model by Sarah Newman, but the basic importance of these structures was first established by Sarah Newman working on mating behavior in hamsters and Don Pfaff, and they were working independently at that time. Andy and I found very similar circuitry in the fish of midshipmen. So the diagram he showed before didn't show all the connections that we mapped out, but you see very similar circuitry in the midshipman brain. And this stuff is extremely conserved in amniotes, and not just at the level of individual nuclei that you can identify that are homologous, but even at the subnuclear level you can get extreme similarities. So this is one of my favorite examples, the lateral septum. The lateral septum of rats can be broken down into 20 different chemoarchitectonic zones, so based on different patterns of chemical innervation, gene expression, etc. We use 10 different markers in birds and can pretty much go from rats to birds and say this is this, this is this, this is this. We didn't break it down into as many zones. We didn't have the exact same suite of markers, but still extraordinary similarities, and comparisons with lizards and chemoarchitectonic work that's been done there suggests that this isn't convergence, that this is conservation. So what we wanted to do was focus on that network and see how that network functions in relation to species differences in sociality. And what we did was purposefully and meticulously construct the most boring behavioral paradigm known to humankind. We put animals in a quiet room, acclimated them, then set it up so that they could look at a same-sex individual through a wire barrier without it eliciting much behavior. Because we didn't want to know that if a violet-eared waxbill got in a fight and a zebrafinch cuddled, that things happen differently in the brain. That's not too informative. But this allows us to simply look at what goes on in the brains of these birds when they're perceiving a same-sex social stimulus, which is something that the gregarious species are normally attracted to and the territorial species aren't. So we waited 90 minutes after that and sacrificed them for analysis of immediate early gene expression. The 90-minute time point here is very important, and it will carry on through the immediate early gene experiments that I described today, because at least in mammals, 45 minutes is the half-life of the FOS protein. You can still see elevations at 90 minutes, but you can also detect decreases when transcriptional activity has gone down in neurons. Throughout that network that I showed you before, we see differences everywhere that map on, that differentiate gregarious from territorial species, that are activated in gradations, that go from territorial to modestly gregarious to highly gregarious, etc. And one of those is a bed nucleus of the stria terminalis. And that's The bed nucleus and the lateral septum is where I want to focus most of the rest of the talk. As far as neurochemical systems, I want to primarily focus on the nonapeptides. So these are vasopressin and oxytocin. In mammals, there are homologous peptides, such as vasotocin. In birds, this is a vasotocin neuron expressing FOS. Nonapeptide systems, in terms of gross anatomy, are very similar across all vertebrate species. The bed nucleus cell group has evolved only in tetrapods, so it's absent in fish. Nonetheless, you see a lot of quantitative variation in the peptide anatomy, and you even see qualitative variation in receptor distributions. So even closely related species may have qualitatively different receptor distributions, which means that they can take the incoming signals and modulate things in a species-specific manner with that signal. This is a completely phylogenetically incorrect way of looking at this, but it's simpler than mapping it onto a phylogeny. The thinking is that early vertebrates had a single nonapeptide, arginine vasotocin, the gene duplicated. Fish express isotocin and vasotocin. All sarcopterygians, except for eutherian mammals. This is a bit of a generalization, but captures the big picture, express this oxytocic peptide called mesotocin. All vertebrates express arginine vasopressin or vasotocin, except for eutherian mammals, which express either arginine or lysine vasopressin. Interestingly, another form of oxytocin was just found in New World primates, all of which seem to express an 8-proline form of oxytocin. So these neurons are expressed in a high density in the bed nucleus of the stria terminalis. They're weakly immunoreactive in a lot of species because they don't hold peptide much. That's not the case in the finches, particularly in zebra finches. They're found in virtually all tetrapods. This circuitry, these neurons are projections up into the lateral septum and other parts of the basal forebrain. It's one of the most seasonally variable testosterone-sensitive systems known in the brain. It's also one of the most ubiquitous plastic features in the brain. It's also highly sexually dimorphic in most species, with males having dramatically more than females. And at the end of the talk, I'll give you a hypothesis as to why that might be. Many species have been looked at in relation to this, but nobody's ever been able to explain the presence of these characteristics. In these semi-opportunistic and opportunistic finches, conveniently, they leave this stuff on year-round. They never shut it down like the other species, which makes it very nice for studying the circuitry. This stuff innervates multiple places in the basal forebrain, the lateral septum very strongly, where vasotocin and vasopressin influence anxiety-like behaviors, aggression, social recognition, and at least in prairie voles, and this is the only case in which we know this actually, pair bonding behavior by acting within the ventral pallidum. So we wanted to know whether these neurons respond to social stimuli similarly in all species, and I'm just going to highlight 2 species quickly for you. The first, the zebra finch. What you have on the y-axis is the percent of those vasotocin neurons that express FOS in a handle control condition in the solid bars, or following exposure to the same-sex conspecific through a wire barrier like I described before. And the activity of those neurons skyrockets and doesn't differ between males and females. You get an absolutely different picture when you go to the territorial species, and I'm highlighting here the violet-eared waxbill. This is a handle control condition. This is what you see 90 minutes later following exposure to a same-sex conspecific. The transcriptional in these neurons essentially shuts down when they're exposed to a same-sex conspecific. We also expose them to their pair bond partner, who they'd been split up from for 2 days. And the activity in these neurons, the FOS activity, skyrockets. So we got to looking at this and thought, well, maybe these neurons are sensitive to the valence of the stimulus, whether it's something positive and affiliation related, or something negative that drives aversion or attack. And in fact, from neurophysiological studies in mammals, particularly primates, we know that other parts of the amygdala are highly sensitive to valence. You have intercalated cell populations that code valence in different ways. You can change a stimulus, change the valence of the same stimulus, and show that the neurons are tracking the valence of the stimulus and not the characteristics, the physical characteristics of the stimulus itself. So we thought, well, maybe that's what we're looking at here, are cells that are tracking valence. So we did another experiment to kind of nail this down a bit. Sorry, I went the wrong way. This is the only picture I took myself, and you can see why I don't take more pictures. But what you see here is one highly aggressive male zebrafinch yanking another male off a perch by his head. Now, we've taunted these birds into doing this. What we've done is we've taken birds, put them in same-sex housing, and then taken two members of one sex and exposed them to one member of the opposite sex. And what they do is they start pushing and shoving over who gets the girl or who gets the guy. Now, normally, this is pretty sedate. Everybody, gets to court. Anthropomorphically, it looks like everybody gets to have a good time. But if you've ever kept groups of zebra finches, they're like kids on a playground. Most of them behave okay. They're a little rowdy, but they're okay. But then there's the bully. There's the jerk that goes around beating everybody up. So we figured, what would these guys do if we taunted them in this way? And they go absolutely insane. This is one of those guys right there. This is not normal behavior for a zebra finch. So we have a group of birds that were handle controls, a group of birds that were, just normal pushing and shoving, but everybody gets to court. And then we have a group of birds that were aggressively attacked or displaced 71 to 205 times in a 10-minute period. And I'm anthropomorphizing, but I don't think they had a good time. So these birds have been in breeding cages before we started doing this, and we've since learned that unlike other vertebrates that shut these neurons down, they stop making the peptide, these neurons are modulating their constitutive levels of FOS expression. So not typically the way we think about FOS operating, but they in breeding contexts, upregulate this stuff, and we know that it's hormonally regulated. The subjugated animals didn't go down significantly, but nonetheless, they certainly weren't going up, despite the fact that they had to have been highly socially aroused, right? These guys had to be very aroused. So arousal is not driving FOS expression in these neurons, whereas the guys that weren't subjugated, over 80% of those neurons are expressing FOS. So we got curious as to whether these neurons did anything that was selective in terms of social behavior, or whether they responded to all kinds of good stuff in the animal's environment. Zebra finches are desert birds. They love to take baths whenever you give them to them. So we kept all the birds, 3 groups of 9 males, on sipper tubes for a month. Nobody had a bath for a month. Then we had a handle control group. 9 birds were given a water bath. 8 jumped in immediately and splashed all around. We put the other one out of the experiment, and then the others got to court a female. Again, before we started this experiment, these birds had been in breeding cages. Baseline's not quite as high as what I just showed you, but still fairly elevated. Absolutely nothing with the water bath. They go up significantly, again, right around 80% when they get to court a female. If you look right next door in the medial preoptic area, and this is just FOS, what you see is that the bath and the female are equally effective. So this bath stimulus will drive those neurons or can drive gene expression, but it's not doing so in those vasotocin neurons. So these neurons are sensitive to social valence, and at least based on the modest amount of experimentation we've done, it looks like they might be sensitive fairly selectively to social valence. We don't know at what level of processing that sensitivity arises. And I just put that in there a little while ago because I always get that question, we don't know yet. So we don't know whether the valence information is derived earlier in processing and is communicated to these neurons. All we know is that the way they behave reflects the valence of the stimulus. We've recently found similar things for mice. These are male mice, handled controls. Males have got to copulate. Those neurons like copulating. If we put males together, if they non-aggressively chemo-investigate the whole time, they activate a little bit, nothing like this. If they go on to fight, it doesn't matter whether they're dominant or subordinate. The subordinates are a little lower, but nothing else happens. So These neurons seem to respond modestly to chemo investigation and very robustly to copulation. Basically, that same design has been replicated now in roosters and with the exact same result. If we look in the lateral septum where those... Did I skip a slide? I think I did. Nope, okay. If we look up in the lateral septum, and these are autoradiograms, we're really zoomed in here so they're grainy. What you see is that a V1A, iodinated V1A antagonist binds primarily in the dorsal or pallial portion of the septum in the 2 highly gregarious species, primarily in the subpallial portion of the septum in the 2 territorial species, and not much binding at all, and in the modestly gregarious species throughout the pallial and subpallial compartments of the septum. So recently, Rilling and colleagues published this paper in Psychoneuroendocrinology, and what they were doing was giving men intranasal administrations of vasopressin and testing them in the prisoner's dilemma game. And what they found is that vasopressin facilitated cooperation with partners when the partner had already cooperated with them. So increased cooperation in response to cooperation. And that was associated with a very selective area of activation. And based on the resolution, you can't say it's in the dorsal septum where the gregarious species have those V1A-like binding sites, but it's certainly covering that area. So this might be a fairly phylogenetically widespread mechanism for driving affiliative behaviors. So all these observations suggest this hypothesis that this circuitry from the BSTM going to the lateral septum promotes gregariousness. And so we tested that in a paradigm that's really simple but works well. The subject's put in a meter-wide central cage. There's 7 perches. The 2 side perches are just a couple centimeters from the cage wall. Zebra finches spend virtually all their time pressed up to one wall or the other next to conspecifics. There's 2 at one end, 10 at the other. This is counterbalanced across subjects, of course. And so we have 2 primary measures that we take here. One is how much time do they spend at the 2 ends combined. That's our measure of contact time. And the other one is the percent of that contact time that they spend next to the group of 10 birds. So that's gregariousness or group size choice. I'm going to show you data from a couple of different experiments. In the first one, we went down with a bilateral cannula and infused vasotocin antisense oligonucleotides into the dorsolateral portion of the BST, as shown by the green ovals. In the other one, and that was a between subjects design, so scrambled oligos versus antisense oligos. And in the other one, we went in unilaterally and infused a V1A antagonist into the lateral septum, and that's a within subjects design, so counterbalanced order of saline trials and antagonist trials with a couple days in between. So we took pre and post behavioral measures in the antisense experiment. So this is post minus pre. What you see in the animals getting scrambled oligo infusions, they actually become more gregarious with repeated testing. The red is the median here. The antisense animals get less. The median difference here is 80%. It's a pretty whopping difference. And that number will come back here again in a second. Counterintuitively, we saw a slight increase in the total amount of contact time. There's much more modest, 25%, but we'll revisit that here in a second as well. We also measured anxiety-like behaviors, and these kinds of assays haven't been as extensively used in birds. They're widely used in mammals. We used a couple of things that are standard in mammals. This one over here is a novelty suppression of feeding test. So if you pair food in the morning when they're ready to feed with a novel object, it increases the latency with which they feed. And what you see here is that the antisense animals, virtually none of, well, some of them did feed, but the median number didn't feed at all. Exploration is also substantially decreased. So this suggests that these vasotocin neurons in the BST are modulating kind of anxiogenic tone or anxiety tone and are reducing anxiety. Now that might seem surprising given that what I said before is that these neurons seem to be fairly selective in terms of what they respond to, but they do have an extremely high level of constitutive FOS expression, so we can hypothesize, and we don't know this, that they're giving rise to a lot of tonic modulation in the brain outside of social context. With the antagonist, this is within subjects. Again, an 80% decrease in our measure of gregariousness with infusions of the antagonist. Absolutely no effect on social contact. So either what we saw with the antisense was a spurious result, or it is being mediated via projections or receptor types elsewhere, and we don't know the answer to that question yet. Latency to feed, nobody fed with the antagonist. So same picture that we got from the antisense. So the vasotocin neurons are responding to positive social stimuli, and they're also reducing what we could call an anxiety-like behavioral state. Now, in rodents, septal vasotocin, or vasopressin rather, is typically found to be anxiogenic. So our hypothesis, and something we're working on right now, is the extreme gregariousness that you see in zebra finches might be associated with shifting this from being anxiety-producing to anxiety-reducing. We know that in the astrilidids, we're getting a shuffling of receptors across subcompartments of the septum, and so that's just a hypothesis that we're currently pursuing. However, in females, the picture is different. You get this whopping effect in the novelty suppression of feeding test, but absolutely nothing on the social contact and gregariousness measures. But that does not mean that septal nonapeptides are not involved in females, and that's what I'm going to talk about here. These oxytocin-like receptors, VT3 and mesotocin. So I don't know what happened to my color here. So this is an older study, so we didn't display the data quite the same way, but it's the same test. On the top, you have the percent of time with the large group. On the bottom is the percent of time with the small group. There's absolutely no effect on overall social contact. It's all about group size choice. This, on the left, is with the subcutaneous administration of an oxytocin antagonist. On the right is with intracerebroventricular infusions. And what you can see is that peripherally, in females only, we see a decrease in time with the large group, an increase in time with the small group. When we move centrally with it, you pick up the males too, so both sexes decrease their time with the large group and increase their time with the small group. If we go into the lateral septum and infuse this locally, again, you get that same kind of effect. So we compared the densities of oxytocic binding sites across all 5 of these species. This is the pallial septum here, and this is the subpallial septum here. This is a zebra finch, blue waxbill, and then the violet-eared waxbill, which is one of the territorial species. So the territorials are in blue here, the modestly gregarious is here, and the 2 highly gregarious are here. So the highly gregarious and gregarious, all the flocking species have more binding sites in the dorsal septum. This tends to reverse. The p-values here is like 0.06 in the ventral septum. And this suggests that modulation across the subnuclei of the septum might be an important determinant of grouping behavior. And in fact, if you express these things as either a ratio or a difference measure, you very cleanly differentiate the territorial from the flocking species. Okay. I want to move on here to talk about seasonal differences and see whether we see similar types of things changing seasonally as animals change their behavioral patterns. I'm going to start off with these 2 species of emberizids, field sparrows and song sparrows, and we're going to bring in a couple more here in a second. We're doing 3 lines of studies with these birds right now. I'm going to only talk about the first one today because it's the only one that's really near completion. And that's an immunocytochemical study where we're looking at 6 different peptide and transmitter systems across a lot of different brain structures, looking at how they fluctuate seasonally in terms of density of innervation and cell numbers and things like that. We've also got a comparable binding study going with peptides, and so that's an autoradiography study. And then I have a student in the field right now working with field-housed animals in winter. They're captured in winter flocks they're in winter flocks in the field, and she's looking at group size preferences and social spacing and dominance in flocks with those birds. But I'm going to focus on the immuno side of this, so I wish we had the receptor side to compare with what I just told you. But even so, what emerged was really quite striking. We don't see this kind of pattern just everywhere in the brain and with every neurochemical system. We see it with mesotocin, and we see it with CRH, and in exactly the same spots. The medial amygdala and the dorsal part of the lateral septum. So this is a field sparrow, territorial in the breeding season, flocking in the winter. It skyrockets. Song sparrows, which are territorial across most of their range year-round, including where we caught these birds, no change. You see a similar blip in the subpalliar or ventral parts of the septum, but not nearly what you see here. And there's a big seasonal shift in the distribution here. I should say that song sparrows, even though they're territorial in the winter, are not as territorial as they are in the breeding season. So how do we interpret this? There is the problem, and just for purposes of time, I'm not going to show you these data, but that innervation correlates negatively with aggression, at least in the breeding season. So we went out in the breeding season, we started playback on territories, measured the latency, the amount of time that it took them to respond, number of flights over and past the speaker, number of songs, shut things down. That was just for 3 minutes, put up a net, and then took a second latency measure. And principal components analysis of that stuff shows that it correlates negatively with mesotocin here. So this increase that we see in field sparrows might just be shutting down winter aggression. It might not have anything to do with grouping behavior per se. And that's where this second pair of species comes in. We have dark-eyed juncos, which are essentially like song sparrows. They're territorial, then group. Towhees, on the other hand, these eastern towhees are territorial during the breeding season, and then they just kind of loosely distribute during the non-breeding season. You might find a few together here or there, but they don't really flock, and they aren't territorial either. So our predictions would be if the increase that we see in septal mesotocin in the field sparrows reflects flocking, then when we compare winter juncos to towhees, the juncos should show a higher density there than the towhee. If it reflects the decrease in territoriality, then these two should look comparable. And in fact, the junco is significantly, shows significant more innervation there than does the towhee. So what's special about the dorsal lateral septum? This is from a recent paper by Luo et al. In Science, and this is from rats, I believe. What they showed is that contextual information being processed by the hippocampus is activating glutamatergic afferents of the caudal dorsal septum and exciting GABAergic neurons there that inhibit GABAergic interneurons in the VTA and essentially disinhibit the mesolimbic dopamine system. So it's pairing contextual information with dopamine release in presumably nucleus accumbens and other basal forebrain structures. And this is something that we want to work on a lot more in birds. We know that the basic circuitry here exists in birds. We know that from some very nice tracing work from Atoji and Martin Wild. If we go back to the ventral tegmental area, what we see is that in the caudal part, interestingly, courtship behavior in the rostral part correlates with activity of the interneurons, not with the dopamine neurons, but caudally, the percent of TH neurons that express FOS correlates with courtship behavior, singing in male zebra finches, and also back there we find that the three gregarious species have significantly higher densities of neurons than do the territorial species. Thus far, we've only gotten a chance to look at the field and song sparrows, but the field sparrows have a significantly higher density of neurons back there year-round as compared to the song sparrow. As I'll show you in a second, though, field sparrows are less aggressive than song sparrows, and this cell density back here correlates with aggression. So before wrapping up here, I want to talk about vasotocin circuits, where we started, right? We know that vasotocin circuits from the BST going to the LS increase group size preferences in the astrilidids, but what about these seasonal birds? Most birds and other tetrapods are shutting this circuitry down in the winter, and we thought, well, maybe they'll leave a little bit more online. And the thinking is this stuff is on so much in the breeding season because it relates to sexual behavior, perhaps, or parental behavior. We know that septal vasopressin is important for paternal behavior in rodents, etc. But what we found was just a total collapse. Very dense innervation here in the breeding season. Totally collapses in winter. The paraventricular cell group doesn't change at all. And so that's what you see over here, summer and winter in a field sparrow. Something else you'll notice, though, is that there is a really big species difference here, enough to drive a species by season interaction. And this relates to aggression. So these are those behavioral measures that I told you about before that we took during the breeding season. This is principal components with both species combined. The latency measures load very highly, so the direction of this PC, the main PC, which accounts for 64% of the variation, is counterintuitive. So low aggression is up here and high aggression is down here. The values for field sparrows and song sparrows are not only significantly different, they're completely non-overlapping. These species are occupying different ends of the principal component. And when we look at the optical density of vasotocin in the BST, we get a very tight correlation with that. So less aggression up here, and really quite a nice relationship. Well, does that really relate to, since we're talking about individual and species differences here, does that really relate to the evolution of territoriality? Well, one thing to remember is that these neurons in most species are sensitive to testosterone, and we know that fighting increases testosterone, right? But this is going the other way. The song sparrows are the ones fighting more, not the field sparrows. So this probably does relate to evolution of territoriality and not experiential differences in the field. And in fact, to breeze over this, we know from previous experiments, this is from my graduate work actually, that intraseptal vasotocin infusions decrease territorial aggression in field sparrows. We did that in enclosures on their natural territories. We know that vasotocin increases immediate early gene expression in the lateral septum, and we know that that immediate early gene expression is correlated negatively with aggression. So to conclude then, in astrilids, evolutionary convergence might not be the right word there, but in divergence and grouping is mirrored in nonapeptide receptor distributions. BSTM vasotocin neurons and septal nonapeptide receptors, using two different ligands, are essential for gregariousness in zebra finches. Septal and amygdala mesotocin, but not vasotocin, track seasonal changes in flocking and territoriality in the sparrows. And the BSTM vasotocin in these seasonal birds, which isn't stable like it is in the opportunistic birds, likely contributes to differences in territorial aggression. And then I have a but here, and this is kind of one final idea that I wanted to throw out there. So you see this big sexual dimorphism, which we didn't quantify here, but you find it in just loads of species, dozens of species. And you see the big seasonal variation in dozens of species. So what's the function? People have just again and again and again and again and again described it in this species and that species and this species, and nobody has really ever gotten to the function of it. Geert de Vries actually proposed a hypothesis some time back that we were thinking about sexual dimorphisms in the wrong way. We tend to think that they promote sex differences, when in fact he thinks that a lot of them probably compensate for other sex differences. So what do we know? We know that these vasotocin cells respond to affiliation-related stimuli. That seems to be phylogenetically ubiquitous, and that septal vasotocin decreases aggression. So these things are up and breeding. We know they're important, or at least in the lateral septum, we know vasopressin is important for paternal care. We know in prairie voles they're important for pair bonding. They're activated during affiliative interactions. So my hypothesis is that the sexual dimorphism is compensating for higher levels of aggression in most male vertebrates, and that this dimorphism offsets male aggression, and it's expressed seasonally because only during breeding for most species are they in a context where they need to not be aggressive towards mates and towards offspring. And that's just a hypothesis, and it would take me about 2 seconds to shoot a couple holes in it, but it's, I think, a good working hypothesis place to start, and it's something that we hope to start working on pretty soon. I'm not going to give you time to read all this, but my lab has always operated very heavily on undergraduate power, and virtually nothing that I've shown you today would have been possible without undergraduates and full-time research technicians. I haven't been very good at getting grad students. I have to pay people to hang out with me. And these people have all been wonderful. Sarah's been with me now for a number of years, and it's very important for a lot of the more recent stuff. Jacqueline Ho did the mouse work with me. Leah Wilson is doing the sparrow work with me. James Klatt did some of the dopamine stuff, and he's also doing some pair bonding work and things like that in finches. Aubrey did all of the antisense stuff that I showed today. Marcy is a developmental neurobiologist that has primarily worked in mice. George was talking about some of her data in his talk today. But she's retooling and working in birds and having a good time. But she's been working with me on and off for a lot of years. She went to Africa to help me start this research program and collect birds. Rick Thompson has been very instrumental for the molecular sides of this work as well. And my sources of support. And I like to end with this these days, and I hope you got one of those out of the talk. Thank you.